Thank you all for the invitation. Before I start, I just would like to know a little bit to whom I'm speaking. So in terms of background, who would think that their background is primarily in biology in terms of studies? Okay, good. Who has at least some background in, say, computer science, physics, mathematics? Okay, so it's a very good mix. Very good. Don't worry at all. I just wanted to get a bit of you. Who has some completely different background that I didn't mention? Good.
Clinician.
Clinician. I apologize for not mentioning this. We do work with clinicians, actually, as you will see. Okay, good. So I think you probably already had a couple of quite intense days and interesting lectures, and a lot of, I think, what one is doing with data in general and also with genomic data is, of course, trying to recognize patterns in these data. So what do they tell me about different types of things? And I think there will be more of this in the rest of the week. Today I would like to take a slightly different perspective. Of course, in some sense it will mainly be about genome sequencing data, as you will see. But of course, if we think about it, in many cases, what shapes the data that we look at, and this can be genome sequencing, then transcriptomic data, data on genome organization, and so on, many of these are in fact processes that are sort of going on. And in my talk, the process that we will be looking at will be processes that conspire to give rise to evolution. Okay? And one of the themes in the talk will be we will be able to measure data at particular snapshots in time. And a particular important snapshot, say for cancer, for example, is someone is presenting with a cancer that is being diagnosed. Often that can be immediately resected, but of course there's a threat of thing recurring. So can we sort of learn something from the actual resected tumor about the process of evolution that led to this tumor, and can we use this knowledge to predict something about the future development as well? So we will try to use these genomic sequencing data to learn about underlying processes of evolution. Okay, so just a brief reminder of what we talk about. Of course, evolution, set the clock running. Evolution, of course, in the sense that we talk about it, came into the world through Darwin and then also some of his contemporaries and development since. And it's really mainly the evolution of species that we talk about. And typically we view this, I mean, these are a famous drawing by Darwin that you all know, so these kind of finches with these different beaks here, that are adaptations to different types of environments and types of food that have been offered by the environment. And of course, this is really just sort of really undergraduate stuff, but let us just recapitulate what we need here to get evolution. First of all, of course, we need variation in the population, and this variation needs to be in a reproducing population where the variability or the variation is heritable. Okay? Otherwise, there's no chance of getting any type of evolution. Then something needs to act on selecting this or that type of variation in the population. We need selection. And of course now the question is, how does the variation come about? And this has for a long time, of course, in evolutionary theory been a big question, very much at the beginning. Of course, in the first half of the 20th century, this was completely resolved. The variation is due to genetics. And this is really essentially by random mutation. Of course, there are other processes that play a role. We can talk about epigenetics and so on, but these are really the basics. And just to give you a number, because this number, and many of you will know this number, of course, but just to be on the same page, this number will be interesting when we then come to our subject of cancer evolution. So if you look at the human genome, well, maybe that's an interesting question to engage. How often do I have a single nucleotide polymorphism in the genome compared to, say, a reference genome? Do you know roughly?
Sorry, 1%.
1% of what? Of the total genome. Okay. I think that's also the number I know. It's 10 to the minus 3, actually. But that's roughly. That can vary a bit, but it's on the order of 10 to the minus 3. So that means if you scale it up to the haploid genome, you have several million differences between any random type of individual. And of course, this type, because this will be, I will not talk about this at all. I will just mention it because a lot of it, what we're now doing with modern data on somatic evolution and cancer evolution is really informed by mathematical approaches of these people that either worked in the first half and the center of the 20th century or later. Kimura was a generation later. And these were really sort of mathematically oriented people who wrote down sort of quantitative ideas about how species evolution operates. And the very important point here is, of course, that this really has led to very productive interplay with data. Okay? All of you have heard about whatever, the African Eve, and that we all go back to this one woman in Africa and so on, and when this took place and so on. I mean, this is really all based on the mathematical theory of population genetics and many more things when we study evolution. Okay, so that's just a little bit about species evolution. Interestingly, that's not typically taught in normal biology courses, and that's probably not because it's not interesting, it's not about biology, but maybe because traditionally it's too maths heavy. But I think this is also changing nowadays, and also thanks to people like you. So what about now, about cancer evolution? Clearly is something that happens in our own bodies. And so do we have the same features there? And just to point out, what do we need? We need reproduction, a population of reproducing individuals. We need variation, and we need selection. So do we have that? And let's just look at for just example, for the blood-forming system. We can look at any other regenerating tissue. It was probably quite interesting that in a young child, very young child, an infant, basically all the bone marrow contributes to hematopoiesis, is full of hematopoietic stem cells. They withdraw a little bit and retreat in the adult out of the extremities. But anyway, we all do blood a lot, and we produce on the order of 10 to the 11 blood cells per day. For the humans, it's not so well known what the real stem cell number is. For the mouse, it's probably you have about 10 to the 4 hematopoietic stem cells and produce 10 to the 8 mature blood cells every day. So it's a massive production out of a relatively small number of cells. Of course, that goes by a large cascade of amplifying progenitors. Okay, so clearly we have a population of reproducing individuals in all regenerating tissues, which are mainly the stem cells. Okay, so and they self-renew by stem cell division, and they also get lost from the stem cell pool mainly by differentiation, but there can be other processes that lose stem cells. Simple cell death, for example, also happens to stem cells. So we do have population dynamics of individuals. Do we have mutation? And of course, if you go to the early years of cancer research, a lot of focus has been on external sources, UV light, smoking, and so on. We know this quite well. But of course, the question is not relevant for the blood, for example, at least not as relevant as it would be for the lung or the skin. Epithelia that are in contact with the external environment. Our stem cells sit tucked away in the bone marrow. But of course, now we know there's a lot of endogenous mutations going on. And this picture has really changed through the last, say, 10 years, in particular also through work by several groups at the Sanger Institute. I would really like to mention here Inigo Martin Corena and his group, who did very important contributions to this, but also many, many other people, but just to single out Inigo. And the picture that has emerged from this is, and again, I guess maybe for many of you this is already good knowledge, but let's get on the same page, is that historically one thought that the endogenous mutations happen mainly as errors of DNA replication. Okay? And this is still an interesting question. But now we know it's not really the copying error that makes the bulk of the mutations, but rather it's actually chemical attacks to the bases in the genome. And they may not happen during replication. They can happen through the whole interface of the cell cycle. And this is large. This is probably the bulk of the lesions that is produced. We may still need, as we will see in a minute, we may need activity of DNA polymerases. We need some element of replication to make these errors into a true mutation. Okay, as we will see. And probably one of the most common is deamination of cytosine or deamination of methylcytosine. And deamination of cytosine leads to uracil, deamination of methylcytosine leads to thymidine directly. So you get this kind of mismatch here. From C to G, you get to a mismatched position here in U to G. And then, of course, two things can happen. Either you repair it, and if you repair it, you're fine, you're back to C to G. Or you actually replicate before you repair it. Okay? And if you replicate before you repair it, you pair your uracil with an A. And if you replicate again, you pair it the A then with a T in one of the daughter strands, of course. And in this way, by creating chemical damage to the DNA and then not repairing it and going through replication, then we get what we would call a mutation. And the typical mutation here would be a C to T substitution. Okay, that turns out is the most frequent single nucleotide variant in the substitution in a normal human genome in most cells. Okay, and this is really work from Ineo's group, I think 2021. So it's relatively recent still, where they devised a method to estimate the average mutation burden of a cell in the population. So it's not a single cell method, but it's also not bulk sequencing, which doesn't give you this information. Okay? Sometimes people, I mean, this is really just an aside, sometimes people report, for example, they did bulk sequencing on this tumor, whatever, 60x, and they have this and this burden, total burden of mutations. I hope after we went through this lecture, you will understand that this is a very problematic thing to say. It's actually quite a meaningless number. That depends on many, many things that people don't think about, and we will understand why. But this just as an aside. So just pure bulk sequencing doesn't help you. Single-cell sequencing is, you have too many dropouts, so devise the method basically to mutation sequence individuals' DNA strands, and then through this get an average mutation burden of individual cells. And that's probably pretty accurate. So I think there's been quite a number of studies now, none of which seriously questions these results. And here we look at hematopoietic stem cells. In humans, stem and progenitor cells, but also mature granulocytes. In humans of different ages here, all these data points are different individuals. And what you see is more or less that there's more or less a linear increase throughout life. This is in a tissue that's renewing, okay? So there is a clear cell division going on. But interestingly, and this caused quite a bit of stir here, there is a very similar rate of increase. It's not identical, it's cell type specific to some extent, but as order of magnitude is seen in neurons that are post-mitotic. Okay? So there's no replication on the genome level there anymore. Of course, there is replicative activity because some of these DNA repair pathways that repair these kind of lesions that are introduced here, they cut out parts of the DNA and then resynthesize parts. And of course, through this, you can also get replicative activity that you need for making these substitutions here. So if you look at the numbers, okay, this is a couple of hundred here at midlife. So compared to the whatever several million SNPs we have, probably 2 stem cells just randomly drawn from the bone marrow of the blood would differ by 100, 200, 300 variants for an adult individual. Does this have any consequence? So we have population dynamics replicating, reproducing populations. We have mutations. We have a continuous ongoing process of mutation that we can actually use as a clock, more or less. I mean, you see very, very nice constant rate here in time. Does it have any effect on selection? And of course, you could easily have selection in a population of this kind if the mutation you get either lets the stem cell renew more often than the unmutated counterpart, or lets it be lost less often, for example, differentiate less often. Both of these would lead to a selective advantage. We don't really understand which it really is in vivo. I think there's intense studies on this, but it's very clear that most of the mutations, most of the single nucleotide variants in the genome that we see, that we still see in the genome, of course, has to be said. So if they were really detrimental, we would probably not see them, are neutral, or at least close to neutral as much as we can measure them. Okay? And this estimate with 99.99% is sort of a little bit from our own work when we estimate the rate of driver mutations in different cancers. It's probably a very conservative estimate. I would expect even more of the mutations are neutral. That means we are, in fact, are now, when you know a little bit something about the neutral theory of evolution in species evolution, where also Kimura made the observation that probably the vast majority of mutations, although they can reach fixation, are actually selective neutral. We are in a very similar situation here. We have a sea of mutations that have no impact on the physiology of the cells, but they are, of course, as it turns out, very informative because they give us, because there's so many, they will give us fantastic statistics on this very small percentage of mutation, really tiny fraction of mutation, that very rarely in a given stem cell can there be a mutation that gives it a selective advantage. There is also a little bit of literature on deleterious mutations. That's intrinsically hard to study because the cells will not really survive very well, okay? But of course, we will, in the second part of the lecture, we will come to look at copy number changes. And it's probably, from what we can see and what we can observe, the inference that most copy number changes are deleterious is probably correct. And so this is quite a different situation from having single nucleotide variants. Okay, and of course we know that this is a paper from Ben Ewert's lab. There's many other papers now on this. I think this was in 2014, so about 11 years ago, that we actually see these clones that have these type of mutations. Interestingly, in the blood, the leading mutations are all with modifiers of DNA methylation or other epigenetics. And we can get large clones, in particular if we wait for a long time. So in more elderly people, we see more of these large clones. Okay, so selection does happen, and it does not lead necessarily to cancer. In fact, probably in most cases it doesn't, which raises an interesting question, which I will not be able to address here. Most of these mutations are actually these C to T transitions. What's the question? Okay, let me formulate the question and leave you with no answer. Okay, and I think we are facing this question now, and I'm happy to discuss ideas offline, but I think it's really one of the fundamental questions now. Let's give it the background. So there's a very famous paper by Tomasetti, is a sort of a computational biologist, more epidemiologist, and Vogelstein, cancer researcher, who's well known through this sequence of evolution of colorectal cancer. And this Tomasetti-Vogelstein paper appeared, I think, first in 2015. So some of you may have heard of it. And it's this idea that cancer is just simply bad luck. Who's heard of it? Good. It's simply bad luck. So it's sort of, you may just get the wrong combination of mutations in the stem cell, then you have really bad luck, and then you are the one that gets cancer, while the other 99.9% of your cohort or so were lucky enough not to get this combination of mutations. Okay, now through this type of work here, we can roughly estimate mutation rates. Okay? And I just give you a number. Say, we have on the order of 10 to the 9 positions in the genome. Okay, so this is the number of nucleotide positions. We would expect that in a rapidly proliferating population, which are hematopoietic progenitors that, say, give rise to lymphocytes, for example, we have on the order of 1 single nucleotide variant per division. Okay? There is a lot of question in the literature, and it's an interesting question whether really division is the right unit or whether it's per time and so on. But for these populations, division is good. That's what I believe also from our own data. But that doesn't matter. Say 1 per division. How many blood cells do we make from progenitors per day? As a human, 10 to the 11 per day. And, sorry? 10 to the 11 blood cells per day. So we have about 10 to the 13 red blood cells. They live about 100 days, so we make 10 to the 11 red blood cells only per day. So they need to come from 10 to the 11 cell divisions. We make 1 error per cell division. That means the repertoire of single nucleotide variants must be, I mean, there's no data on it, but simply from these data, should be completely saturated. Every single position in the genome, in the blood progenitor cell, should be mutated in one of these progenitors. Okay? So it's not that the mutations themselves are rare. That's the question. So it's not bad luck that occasionally the wrong mutation happens. All of the bad mutations happen all the time. Yeah, and we see them. So so in the blood it's been a bit harder. Of course, we see these things. All of these are pre-leukemic. These people never get leukemia, okay, in their lifetime. Yeah.
Are these patients silent?
Sorry?
Are these patients silent that you show in the slide?
How do you mean silent?
I mean silent.
I know they're not silent. So they are functional.
They're all missense.
Sorry?
They're all missense, right?
No what?
Missense patients.
Most of the DNMT3A are mostly loss of function. Exactly, yeah. So they're all functional mutations, so there's no silent mutations counted here. Okay, good enough. Silent mutations are also not selected.
There's a question there.
Yeah. Yes, sorry. Hi. We're getting away from the subject of the lecture, but it's just completely fine.
Is there a recombination? Does it happen, or it's...
No, I think we also see larger structural changes.
No. I mean between. So here you have asexual kind of clones, basically.
Yeah.
Is there any recombination?
So far, all of these models of somatic evolution do not place a big role on recombination.
But has it been shown if there is?
Interesting question.
So no.
I think it's not, to my mind, it's not entirely clear to what extent this plays a role. Okay? I'll leave you with this unsatisfactory state of things. Good. So, good. But let's first register that question, and for the rest of the lecture, I would like, or for the rest of the talk, and please do keep interrupting me also for questions. I would like to not try to answer it. Happy to discuss it. Okay. But really try to see, can we, just by looking at cancers, can we say something about their path of evolution? Okay, that's all what I want to say now. So it's a relatively simple question. It's rather descriptive. I want to say, okay, I have this tumor. I want to answer simple questions. For example, when did the cell of origin of this tumor arise? Okay. Okay, let's see whether we can do this. So just for the somatic evolution part, which is there's endogenous somatic mutations that accumulate more or less at constant rate, and mutations can become selected. And in many cases, these are sort of pre-malignant type of clones, but in other cases, for the liver, for example, the liver, in cases of metabolic disease, it has also been shown that there can be selection for other criteria, so metabolic adaptations, for example. Okay, so let's go into cancer, let's call it cancer archaeology. So what we typically have, and what we can easily get, and where there's lots of these data, is whole genome sequencing data. And that's also the starting point when we started trying to be interested in this work. It's actually only about 10 years ago, so published our first paper on this in 2019. I think probably started around 2015 or 2014 working on these things, maybe a bit earlier. And I just thought there's this humongous amount of whole genome data of cancers that was initially made to find driver mutations or driver events in the genome. Okay, now we have pretty good catalogs of driver events, and can we use that for something else? Okay, and that's also, of course, simple for you. Let's suppose we have a collection of reads. here that cover these type of positions. Let's suppose we know that they all come from tumor cells. That in itself is an interesting question, but let's say it's a leukemia, we can sort for the tumor cells. And let's say we just look at two neutral single nucleotide variants, one that is in 50% of the cells, or roughly 50%, and the other one is in a bit less, about 10% of the reads, not the cells, of the reads. Okay, so the genome is diploid. So let's suppose the disposition is diploid here. Okay, then 50% of the reads means, because it's a diploid genome, it's in every single cell. Okay, the other read is unaltered from the other part of the genome. That means 50% means these mutations are in all the cells, that what we would call clonal. While these are in 10% of the reads, are in 20% of the cells. Okay? So they're what we would call subclonal. And that's what we typically see in cancers, and we can classify the mutations by this. Now, a very simple thought now comes is that is almost trivial. Well, it is trivial. If these are in all the cells of the cancer, that means they must have been in the one single cell from which the cancer ultimately grew. So they're what's called in the founding cell of the cancer. And often when you read this literature, there's a technical term for it that comes actually from this population genetics theory of species evolution from Fisher and Wright and other people held in. It's called the most recent common ancestor. Okay, and we will occasionally say that's the MRCA. Okay, so that's the cell from which the cancer that I resect grew clonally. Okay, good. And so let's now look at this process. Say I have a normal tissue, okay, and I look at some cell divisions in this normal tissue. So suppose these are stem cell divisions in this normal tissue, and then I will accumulate somatic variants, okay, per division, 1 or 2 or something like this, until I really get a mutation here, the blue one, that gives cells a selective advantage. And then with the selective advantage, a clone grows out, and maybe that's already a tumor. In some simple cases of especially pediatric tumors, even one mutation can be enough. That's very rare, but there's a couple of cases known where a single mutation can grow a tumor. Okay? So and then these will be clonal in the tumor. These will be the ones that I've just seen, okay? Including, so these are neutral, and this is the one that's the actual driver. Both will be clonal. And then, of course, while the tumor is growing, here in this kind of subclone here, I get these kind of subclonal mutations very well, okay? Now, as you can see, I accumulate mutations here. The clonal mutations are accumulated before the tumor arose. Okay, and the more I have, the longer the time span. I have to apologize, sorry. Can I switch that off? Maybe I switch it off completely. The longer the time span was before the tumor started. Okay, so now I can draw my typical histogram of variant allele frequencies. Okay, so all of these mutations will have a variant allele frequency of 50%, they sit here, and there's more or less binomially distributed. It's a bit broader than binomial, but roughly binomial, because I draw randomly from reads, from the sequencing reads, and then I have a tail here, and this tail has also a characteristic shape that can be predicted by population genetic theory for these cell populations, and this contains all these subclonal mutations. And as already said, the height of this peak, that's proportional, or the height, the integral of this peak is proportional to the amount of mutations I accumulated in the founding cell of the tumor. So that gives us, in fact, the age of the most recent common ancestor in mutational time. And that's very simple. You can do this for every tumor. It's totally easy to do. Okay, so what we can get from here is a little bit more tricky and involves a bit of mathematical theory, but it turns out that the slope of this subclonal curve is actually related to the mutation rate. So we can also get an estimate of the mutation rate from these data. And I'll come back to what type of mutation rate it really is, but it's an interesting type of mutation rate. It's the number of mutations counted per what is called an effective division. And an effective division is basically a series of divisions that lead to a doubling of the stem cell or the cancer stem cell. And in this case, this would be this first division here, this cell is lost, second division here, this cell is lost, and only the third division really leads to a doubling of what is actually growing the tumor further. So there would be 3 actual divisions per effective division. And that becomes important later on if I have enough time. But the important point here, you can get very simple measures of first of the time of origin, but you can also get something about the process, or in particular of the mutation rate. So what we then do, and which I will not dwell too much on it here, but just give you a little bit of a feeling, is we now write down this process and in the simplest possible terms. Okay, we say we have a stem cell or then cancer stem cell population that has a certain self-renewal rate lambda, has a certain loss rate, which can be a compound of a differentiation and a death rate, and it has a rate at which it accumulates mutations, and these can be neutral, and in some cases, very rare cases, this will be advantageous driver mutations, but they could also be deleterious. Okay, so we know on the other hand, so this is our theory, and we can write down the theory in the way I just describe in a second. These is our measurements. This is really the simplest type of measurement. Of course, one of the avenues to go here is to take these bulk type of, you need reasonably deeply sequenced tumors, so whatever 15x or so will not get you very far, but if you have 50, 60 more, sometimes we have 150 X or so. This is still nowadays is not particularly expensive. I mean, you can get these samples that will give this variant allele frequency histogram. So this is our data. So we want to connect the model to the data. So we write down a bit of theory by saying, well, this is the number of variants with clone size I. So this is any height here in this histogram. Okay. In population, traditional population genetics, this is called the site frequency spectrum. Doesn't matter. And so how many mutations do we get at a certain clone size I here? Okay. Well, this will be the rate at which mutations originate here. So this is this mu times lambda for this process, times the number of cells we had at a certain time, n. So we need to know something about the dynamics of our population of interest, say of a cancer stem, of our stem cell population in which the cancer arises. Okay, and at a certain time point, t dashed, I had this number and this probability to get a mutation. And what's the fate of this mutation in between t dashed and the measurement time t, at which I do my whole genome sequencing measurement here? Well, that depends now on the dynamics, on the clonal dynamics. Okay, so what's the probability that a clone that started with a single cell at t dashed grows to size I in the interval until t? Okay? So that's really a theory of stochastic process here that enters this theory here. So, but in this way, you can write down a closed formula for the site frequency spectrum. Computations will be complicated, but manageable, simply because this is here really a stochastic branching process was enter here, but everything is manageable. You can do that. And you can now write it down, and we really don't want to go from this side to that side, but we want to go the other way. We want to say we have these data, we know what the underlying dynamics are. Can we learn from the data the parameters of the underlying dynamics? And you can do that. And for the sort of main part now of the actual biology, I would like to talk about one particular case study that we did that's already published now 2 years ago. So you can read it. We will also look at this in the tutorial. But it turns out it's a very interesting tumor, and we've done a bit more since, and also other tumors. And in some ways it's quite paradigmatic of a class of tumors that children get. And luckily they're very rare. So it's called neuroblastoma. It's a tumor of the sympathetic nervous system that most often grows at the adrenal gland. So it's at the peripheral nervous system. And as you say, see small children get this tumor, luckily quite rarely. But among the childhood tumors, it's one of the more frequent ones. Yeah? The tumor is very interesting because it has all possible outcomes, and that range from spontaneous regression to fatal disease, and about 30% of the high-risk cases are still fatal, so we cannot help the children. Yeah. But it now turns out, of course, that it's very important to diagnose the child correctly and predict how the tumor will develop, because if you have a good chance of spontaneous regression, you don't want to treat. Right? While if you have no chance and if you have very high-risk disease, you want to treat as strongly as you can to have some possibility to actually cure the child. And there's one very odd observation in this tumor. Although the age of diagnosis is spread out not too much. I mean, there are some older cases also beyond 6 years. Most are between birth and 6 years. That has gone entirely unexplained, but it's used clinically. When the doctor diagnoses the child, they also ask the question whether the child is younger or older than 18 months. And if it's older than 18 months, experience tells this will be more likely to be a critical case. No one knows why. Okay, good. So much about that. So if you look at neuroblastoma genomes, they look moderately disheveled. So you do see you have a lot of copy number gains and some, in this particular case, I fail to see a loss, but you also have losses. Maybe I'm standing too close. But typically it's whole chromosomes or arms of chromosomes. So there are some cases that are chromotriptic, but it's not very typical. Okay? So basically you have chromosomal or arm level gains and losses in these. Okay? So now let's do our statistics for the most recent common ancestor. But now we need to do our statistics, of course, by ploidy. Okay? So let's look first at the chromosomes that are disomic. So that would have the normal copy number that's down here. Sorry, here are the disomic chromosomes. So what we see here is this kind of clonal peak for the most recent common ancestor that we would expect. We would think it should be at 0.5. This is uncorrected for tumor cell content. It tells you that the tumor cell content here is probably pretty high, but it's not 100%, but it's in the range of 80%. Okay? So, but all of these mutations are in the most recent common ancestor of the tumor. We can count them. Okay? Now, do we see this signature also in the other parts? We also see that here. These are the trisomic chromosomes. These ones here. We see this clonal peak here. Okay? But it's now because we have 3 chromosomes, the clonal peak is at 1/3. No longer at 50%, it's a little less than 1/3 because of the tumor purity, and you can go even to the tetrasomic chromosomes at 1/4, as we'd expect. Okay? So now we can count all of them, we can convert them into density, so in mutational time we know when the most recent common ancestor happened. But we observe something else quite interesting, and this is, look here at the trisomic part, we also see a small peak, but very significant one, of not at 1/3, but at 2/3. Okay, 2/3 means it's on 2 of the 2 chromosomes. Okay, so how can this happen? Well, one of the chromosomes in the trisomic constellation obviously got duplicated, and these were the mutations that were present on the chromosome when it got duplicated. Okay, so this density times the time point of duplication of the chromosome. Okay, good, clear so far. Okay, so now let's do a statistic. So now let's convert them into densities of somatic SNVs per megabase pair, and we see some number here, okay, 0.2. And this comes from the aggregate of these peaks. Okay? Now let's look at at this peak here, and we also get a number, and it's much, much closer to 0. Okay? We don't quite see it from here because we need to do it as a density, but really most of the chromosomes really come at this very early time point here. What it's telling us is that most likely these chromosomal duplications that led to the structure of this genome here happened before the tumor grew out clonally, okay? Because the mutational clock has a much lower reading there. Okay? And we can now compare it, of course. So, yeah.
Sorry, maybe it's obvious, but can you explain why?
Because you would say, okay, There's nothing specific about these regions and their whole chromosomes. Okay, so I don't expect that regional differences in mutation rate play a big role in these statistics here. Okay, you can try to do that, and we try to adjust it when we test here. So we try to adjust for some dispersion in the distribution due to regional differences in mutation rate. Okay. So now you look at these pieces that were duplicated and look at the density of somatic variance. And it's whatever, 0.01 or something like this. Okay? So that means this must have happened at a time when there was this amount of somatic variance accumulated. But the clonal outgrowth of the tumor happened at a time when there were 20-fold more mutations accumulated. So most likely the explanation is that it was later. Because exactly. Yes?
If this tumor is mainly copy number driven, why don't you use the copy numbers to estimate
To estimate what?
the tumor fraction?
No, of course, we estimate the tumor cell fraction. I didn't go into this here. And we also use the copy numbers for this and so on. Yeah. So in this particular case, it's probably around 80%. Sorry?
So the number of mutation in these tumors is relatively low, right? It has to be like below 1,000.
Exactly. It's a pediatric tumor from a small child.
So wouldn't it make more sense to just use the copy numbers?
I'm not trying to estimate tumor purity.
I know, but in the calculations that you do, can it be done just like the copy numbers?
I'm using the SNVs as a clock. So my argument is based on accumulating SNVs on these segments that I want to study.
But it can be done also with the copy numbers.
No, the copy numbers are not a clock.
The copy numbers are not closely, they just show the
And I will show you in the next slide why the copy numbers they're not accumulating in a clock-like fashion, because now we can use the, yeah.
Yes, sorry.
I don't want to get confusion. Let's pause here, but maybe your question?
I got confused. Can you please explain us more the difference between clonal on one copy and clonal on two copies?
Okay, so this is one chromosome, that's the other,
Yeah.
one A and B, okay? Now, this gets duplicated now, okay? I cannot draw another one here, okay? So all the variants that were at the time point of duplication on this one also get duplicated. So now so forget about clonal. So basically there are on two copies, and there are two copies in all the tumor cells because this happened very early. Okay, so that's why it's also clonal. Forget about it. So everything we see here, we see no subclones here that are interesting. So the tumor is basically clonal. But this duplication event, everything that was there before is now on two copies. But everything that comes afterwards is on one copy, plus also the things that were here before but were not duplicated also on one copy.
Okay.
So that also needs to be calculated somehow.
Thank you.
Okay, good. And good. So let's now try to summarize what we learned from here. What we also see is that there are two chromosomal gains here that are not mapped to this early time point. Question whether they're significant. They're more closely mapped in mutation density to the time point of the most recent common ancestor of the clonal outgrowth of the tumor. All right? Good. Right. So if we now take this picture together and we put confidence bounds based on some statistics that get these dispersions in on it here, what we see is that most of the chromosomal gains that we can time fall onto an early time point, and they're significantly different in timing from the most recent common ancestor, and it's most likely that this was a single catastrophic event that led to what is called a triploidization of the genome. Okay, so one wrong cell division most likely caused that. Okay, it should be said that several of these gains here are known recurrent drivers of neuroblastoma. So 7 gain, 17 gain, I don't know what's in this tumor, we have to look. So it is very likely that a subset of these gains here have selective advantage, so led to a pre-malignant clone, but then something else needed to happen later on for the tumor to really grow. Question? Oh, good. And these gains here, of course, there's no real confidence on this, but it's more likely that they actually occurred at this time later. Okay, need to watch time a little bit, but we get through. Okay, now let's go through an entire cohort of tumors. So that's the cohort. And we see there's two cases. This is the case we just saw. And there's other cases that do not have a really catastrophic event, but they just have an isolated gain. 17 or 17q is very common. It's a very frequent gain in neuroblastoma that's almost defining, so this probably has a selective advantage. Okay? You don't see the other stuff, so that's why we also think other stuff is deleterious. I mean, other stuff going with 17 can be seen. Okay, later on in MRCA, and now we can, over our cohort here, we can get the cumulative incidence. This is of the early event, and this is of the most recent common ancestor, and you see clear shift in between. Okay, so there's a very early event, and later on there's clonal outgrowth of the tumor through some other event. Come back to this. We have a second class of tumors, and this is now blown up in scale. So this is here, this is this little scale here, blown up. Okay, that only have this first event and never get a second event. Yeah? And in this case, it's also probably a triploidization of the genome. Occasionally, we also see genome doubling. Okay, if you now get the incidence of these tumors here, they have this early event here, nothing else later, so this defines the origin of the tumor. Okay. Defines the origin of the tumor. And if you look at timing, it agrees in timing, in mutational timing, with this early event of these tumors that get a second event. Okay, good. So now we did some statistics over a larger number of tumors by saying what is the mutation density of this MRCA. Okay, and we have these two groups. Maybe I should just say this. This is the group that has an early MRCA, where this early event already defined the clonal outgrowth of the tumor, and this has a two-hit, and that's a two-hit kind of tumor. Then the first hit, and then something evolved, and later it grew out clonally. These I will call late MRCA. These I will call early MRCA tumors. Yeah. And now let's say, indeed, the density is bimodal. We have these early MRCA tumors here. We have the late MRCA tumors here. And we can now try to validate this in tumors that we haven't analyzed that are also more shallowly sequenced. So this is something you could do in a clinical setting. And we see the same picture. We have early and late MRCA tumors. Okay. Now we looked into these. Okay. Maybe I'll show you first this before we look molecular into these. So I said in the intro that it's very important to, when these children get diagnosed, to assign them to the correct treatment. Okay? And one way of doing this is clinical stage. And I will not get into how clinical stage is done. That's a histopathology type of score. Okay? So this is staging of children that were considered to be low risk and staging of children that are considered to be high risk according to clinical stage. So this is the pathologist looking into the microscope. And this is the survival of these. Okay? So you do see, you do well, pretty much, but there's a group here where you do wrong. Okay? So these are actually high-risk cases that were wrongly classified by stage as low risk. Now let's do our evolutionary assignment by saying, is this a tumor that evolved long, so it has a MRCA that comes late, or is it a tumor that evolved only short and grew out quickly? And let's try to link this now to outcome. Okay? And that's what the result is. So that means practically, except for one case, all of the early MRCA tumors are actually better predictors of a low-risk disease than the histological score. Yeah? And the late MRCA, vice versa, a predictor of high-risk disease. So why could this be? We don't fully understand it. But interestingly enough, we're now trying to test this in a clinical trial. So would this hold up if we test a larger number of patients? So could we, just from the duration of tumor evolution, infer something about how aggressive the tumor is? Good. So let's look at some, again, descriptive molecular features of those. So if we now look for the early MRCA tumors and just see, do they show enrichments in certain features? What we notice is they're depleted of these stage 4 tumors that are histopathologically high risk. They have whole chromosomal gains or losses. and particularly here, these are typical oncochromosomes for the neuroblastoma, chromosome 7 also for many other tumors. And they typically also have this near triploid genome I showed at the beginning, but not exclusively. Some of them also have that. While almost without exception, the late MRSA tumors, they have this early event that consists of chromosomal gains, but already the chromosomes break at these early events. So they have some kind of segmental gains or losses, so they have some kind of chromosomal instability very early on. We do not understand why, but this distinguishes these two. And again, these have poor prognosis, they have good prognosis. Okay? What we also see in these tumors is that they get the second event they usually get at their most recent common ancestor is a mutation that leads to a gain in telomere maintenance. And there are different ways of doing this molecularly. In neuroblastoma, there are 3 different ones. I skipped it to not complicate two things, but there are 3 characteristic mutations that lead to a gain of telomere maintenance. Okay? What could that mean? And these tumors never have that. Okay? That's also characteristic. Good. And incidentally, I said this at the beginning, of course, they evolved longer, so they're diagnosed later. And this agrees with this age over 18 criteria.
May I ask a question?
Yes.
When you say, what is the late MRCA? If you have fast tumor in which you have fast clones with high selective advantage and so on, then this would create a late MRCA, right?
No. Yes, of course, you're correct. So if we had a situation where there was a lot of subclonal evolution and one subclone would actually really take over, this would create a late MRCA.
But
correct.
this is the distinction between these tumors and no.
The interesting thing is that so we don't really know what happens in between. So this is also one of the things we do with this archaeology, okay? But in particular for these type of tumors, there's practically no subclonal evolution when we dissect the tumor, okay? So everything is behind us already. So the last clonal sweep dominated what we see, okay? So we don't really know what's going on here. I will give you one additional thing to think about, what might have gone on. But it's very clear that these tumors here all have this second event, although they had a very similar first event as these, while these tumors don't, they just grow, but they grow more benignly. Okay? So something, you're completely right, something happened here in terms of subclonal evolution and selection that made these tumors aggressive. Okay? And I just want to give one hint to think about why possibly this is the case. And they become aggressive, at least if you look at the type of mutations they get, by having increased telomere maintenance, okay, which is not uncommon, of course. So let's do one more population dynamic thing. I think we're good. We're discussing, and I finish in 5 minutes, so we're all in time. And there will be more in the tutorial, of course, of this. The tutorial will actually be about this example. Okay. So, yeah, sure. About which one?
No, the early versus the late MRCA.
This. Yeah.
So do you think that, how heterogenic are these tumors? Like, could it be that the late MRCA are just heterogenic? So some of the cell type populations have some of the mutations, which is why some of them respond to treatment and the others don't. Well, these are common.
Yeah, that's an interesting point. So I'm happy. It's a very good point for discussion. I give you the answer, but you will not be satisfied because I don't give you the facts. But we can look at the facts together. I think these tumors are essentially clonal when you diagnose them. And of course, there is heterogeneity, that's very clear. But we see no evidence that this heterogeneity plays any role in their treatment resistance. Okay. That may have different reasons. That may also be because our measurements are too limited. So at the moment, we have a very limited set of primary tumor and recurrences, and that's what we need to look at your question. I think.
Also, it would be interesting if you said you had nearly single cell.
Exactly, yes. Yeah, absolutely. And I think there's a lot of to be said for combining these, and we're doing this in the group now also in several projects, for combining these deep bulk data Often people who work in single cell are a little bit dismissive of bulk data, but bulk data are great because they are a representative sample of what's going on in your tumor. Okay? And if you sequence deep enough, you get a representative image. Okay? If you only sequence 10x, you don't. Sorry?
How low can you go with the bulk sequencing?
How low to still see something?
Yeah.
You can probably time an MRCA with 30x. But subclonal structure, I would say, starts to get unreliable at that depth. Yeah. Okay. Also, I think to really get into subclonal structure, bulk sequencing is not really the method of choice. I mean, you see something, if you go very deep, you start to see things. But of course, it's not the greatest method. If you go to single cell, you have this fantastic resolution of the single cell, and you get also co-occurrence of mutations in the single cell. But you should be aware of that your sample is always small, whatever you do. Even if you sequence 10,000 single cells, it will be a tiny fraction of the tumor. And whether the clone that will eventually win is really represented in there is an open question.
But do you believe the CMV estimation of the single cell method?
The what?
Do you believe in the copy number variation estimation of the single cell methods?
I think the copy numbers are great from single cells, to be honest. I mean, I'm happy to discuss details, but that's, we usually believe the copy numbers from the single cells, even the inferred copy numbers from the transcriptomes. So, but there you are. This is an ideal tumor for this because we have all these copy numbers. You could do both. Yeah. I mean, genome sequencing is safe, but I think even from the RNA sequencing you see, and that's one of the okay. I need to finish at some point. That it says I spoke for 53 minutes, so this 7 minutes. Yeah. Okay, I want to finish at some point. I don't get through entirely, but okay. So I wanted to give you a little bit of a glimpse what we think happens in the evolution of these late MSA tumors, and this is now. Linked to one particular way of how we use these population genetic models that I'm happy to discuss. I will not explain that well. So now we take actually this model and say the neuroblastoma evolves from a dividing cell population in which this mutation happens. Okay? And the first mutation will give a selective advantage, and the second mutation even more. Okay? So, and we can fit these models to the data. I will not explain. What we basically fit are incidences. So this would be the early common ancestor incidence. It would be the most recent common ancestor incidence. We get estimates of mutation rates, so we can real-time things. And you see, for example, here, one of the insights that really stunned the field at the time is that we see these early copy number events happen in the first trimester of pregnancy, so long before the child is born. Which incidentally, it sounds terrible, but on the other hand, it would also tell you maybe there's scope for early detection. So, and that's a big issue in this field. I will not speak more about it, but that's what we currently try with a pediatrician in Heidelberg to really try to explore how common is this in other tumors of the pediatric tumors of the nervous system, and can we exploit that? It's a long way to go, but these happen early. These happen typically after the second event, and this happens typically after birth. And what we can also predict, and I'm just saying, is what is the feature of the cell population in which the first event happens? And our prediction is this is a transiently expanding cell populations around the end of the first trimester of pregnancy, and that's a known cell population, and there's no fiddling here. This is really coming out from our model, but from embryology we know that the sympathetic neuroblasts that form the adrenal gland proliferate strongly at this period of development. So it's most likely, and we have other evidence from single-cell RNA sequencing together with a collaborator that sequenced embryos, that these are probably also the population from which the tumors arise. So it looks like that this very early expansion of this neuronal progenitor population creates vulnerability for this tumor. Okay, but to come to the point is what we also estimate here with these models is we estimate mutation rates, okay? And we estimate mutation rates in two different ways. We estimate from the subclonal tail here, where are we? We can estimate what's called the effective mutation rate, namely the number of SNVs per effective division. And this is 100, okay, on the order. So our typical estimate is even 150 here, okay? This for one particular tumor. That's massive. Okay, so but they don't get 150 mutations per cell division. We can also estimate the mutation rate in a different way, which I will not explain here. And this is more what we would expect. It's 1 to 3. Okay? So we have a difference between 100 and 1 to 3. That means there are 100 over 1 to 3 divisions per effective cell division. Okay? Which then means that most of the divisions of the progenitors that lead to the tumor and most of the tumor stem cells in some way will not lead to growth of the tumor. Okay? They will either lead to cell differentiation or cell death. Okay? Interestingly enough, when we approach the clinicians with this finding, they said, well, we believe you because the aggressive tumors are characterized by a very high fraction of cell death. Okay? They look pretty cell death-like. Okay? So, and this is something we find in many, many cases. For other nervous system tumors, that the rate, that the fraction, that the probability that an individual cell division really contributes to a lineage of tumor cell growth is small. Most cell divisions do not contribute. Okay? And if you now look over the aggressive and non-aggressive cases, so these are the non-aggressive ones. This is the cell loss ratio estimated here. It's still pretty high, but it's in a much, much lower range here. I mean, this is above 90% for all the aggressive cases. So what it looks like is that in these cases here that already show early chromosomal instability for reasons that we don't understand, they go through a crisis in cell survival, and they need to develop mechanisms for telomere maintenance, otherwise you wouldn't get a grown tumor. So in some sense, a bottleneck in tumor growth makes the tumor aggressive, evolve into something aggressive. That's what we think. Okay, good. Here we are. We see this for other nervous system tumors. We see, okay, I shouldn't say. We now looked at last idea, because this links into what we did before. We now started to look into normal brain tissue of elderly people who died of other causes. And you would expect that occasionally you would find these initiating mutations, at least of adult tumors, not of the childhood ones, because they're gone. And this is just to show that we do see. So this is an elderly person, not so elderly, but he died of some other cause, a completely normal brain, but he has an initiating mutation of a glioblastoma, interestingly. And also that's, I didn't have time to explain this, that's also in a selected clone. So basically this person has a pre-malignant clone that with one further hit would have possibly become a brain tumor, and when we found one other case of this. Okay, so let's summarize here, and thanks a lot, almost in time, for your attention, for the great questions also. So I think we can time key events in cancer evolution simply from whole genome data, and there are lots of whole genome data to be explored. Okay? Many more than a single group can do, and chromosomal gains can be timed very well, most recent common ancestors. There's an interesting problem that lady in the back, what's her name? Sorry? Maya mentioned is, of course, estimating tumor purity correctly from samples and so on. So there's lots of interesting technicalities. What comes out from this is that in every case we looked at so far, the actual tumor origin is at least 1 to several years before the time point of diagnosis, and in many cases even decades. And other groups have described the same. There's a very good paper by Moritz Gerstung of 2020 where they look across many tumor types, and they also see years to decades before diagnosis. What this means is that, of course, there would be scope for early detection if I could do this more sensitively. Okay. I also showed a little bit at how the dynamics of tumor evolution, at least in this kind of cell death case, can be reconstructed by looking how the patterns of these many neutral variants is shaped by the drivers, right, by the clonal drivers. And I think I already made this point. I mean, we found this for other nervous system tumors now as well, and possibly it also applies to a larger range of tumors. That we see that in the cases that we studied, it looks like that the actual selective pressure for cell survival really was the feature that made the tumors very aggressive. Okay, thank you very much.